Students, welcome to biology class. Today I am going to teach you structure of chromosome part 2. So let us begin. Structure of DNA. This is the full form of DNA, deoxyribonucleic acid. Watson and Crick gave double helical structure of DNA in 1962. So here is the structure which I have already explained you, but let me revise it. As we know that this is the pentose sugar, this is phosphate group, and this is nitrogen base. This is one nucleotide. All these nucleotides join in a parallel sequence, and this way you can see them. In the middle, if you see, both the nitrogen bases are joined by hydrogen bond. They are weak hydrogen bonds. And on the side, we can say backbone is joined by the phosphate diester bonds. Now this is one nucleotide as it is shown over here. This particular one-side chain is called polynucleotide chain or strand. So there are two polynucleotide chain, one and second polynucleotide chain or strand. That's why we say DNA molecule consists of two polynucleotide chains or strand. Now this is the double helical structure of DNA. Let us see what does it says. Now polynucleotide chain, these chains form double helical structure of DNA. The two strands are anti-parallel, that is, one runs in 5' to 3' direction while other runs in 3' to 5' direction. So this is from pentose sugar 5 and ends with the 3, and here it starts with the 3 and it ends with the 5. That's why they are called anti-parallel strands. Next point is the one whole helical structure is 34 angstrom. So the width of the DNA molecule is 20 angstrom. It's like a spiral staircase, as you can see over here. In one particular helix where the turn takes place, it contains 10 nitrogen base pairs. Between the two nitrogen pairs, the distance is 3.4 angstrom. Do remember, students, always purine nitrogen base A will pair with pyrimidine T, and between A and T, two bonds of hydrogen are only present. Purine G always pairs only with C pyrimidine. This type of pairing is called complementary pairing. Dear students, now I will explain some terms related to chromosome. Now let us see what are nucleic acid. There are two types, DNA and RNA. Let us see the difference between them. DNA is formed of double helical strand, while RNA is formed of single strand. Here the pentose sugar is called deoxyribose, while in RNA the pentose sugar is called ribose. DNA is made up of millions of nucleotides, while RNA is made up of only 10,000 nucleotides. Now let us see what are sister chromatids. Suppose these are two chromosomes, chromosome 1 and chromosome 2. Or we can say, suppose this chromosome is parental chromosome and this is maternal chromosome. Then the arms of one chromosome are called sister chromatids, while the arms of two chromosomes are called non-sister chromatids. So this term we are going to use in the next chapter, that is cell division. So do remember what are non-sister chromatids and sister chromatids. Now we are also going to come along with the homologous chromosome term. So what is it? In diploid cell, you know the chromosomes are present in pair. Two chromosomes which are identical in appearance have similar pair of genes throughout their length in the same sequence are called homologous chromosomes. One chromosome is inherited from mother, maternal, by the egg, and one from the father or paternal by the sperm in this homologous chromosomes. So, a human cell has 23 pairs of homologous chromosomes. Now the question comes, what is crossing over? Dear students, these are two chromosomes, maternal chromosome and paternal chromosome. Now here the non-sister chromatids, they cross at one point. This point is called chiasmata, where the crossing over takes place. And this point has a contact between the two arms here. In some of the books, it is also called synapses, is the pairing of the two homologous chromosomes that occur during meiosis cell division. Now, what is crossing over? Crossing over is the exchange of genetic material between non-sister chromatids of paternal and maternal homologous chromosome at chiasmata. Some other terms like diploid chromosome and haploid chromosome also we should know for the next chapter. Diploid chromosome is symbolized by 2n and haploid chromosome by n. When paired chromosomes are present, then we write it as 2n or diploid chromosome. So 2n number of chromosomes are present in all the cells except egg or ova and the sperm. When single chromosome is present or single set of chromosomes are present, then they are called n number. N number of chromosomes are present only in egg and sperm cells. Now what are somatic cells and what are reproductive cells? All the cells throughout our body are called somatic cells except testes and ovaries. And here cells present only in the testes and ovaries are called reproductive cells. Now somatic cells contain diploid or 2n number of chromosomes, do remember. And so somatic cells always undergo mitosis cell division, while reproductive cells contain haploid or n number of chromosomes, and reproductive cells always undergo meiosis cell division. By this I end up my lecture